Welcome to Biochemistry Unfolded, From Atoms to Algorithms, an online podcast on the topic of biochemistry and biophysics. So welcome back to Biochemistry Unfolded, From Atoms to Algorithms. So today we're going to talk about AlphaFold. And in 2021, let's go back to 2021, and Google DeepMind's AlphaFold did something that got like magic. Feed it a sequence, and it hands you back a protein structure, often with just jaw-dropping accuracy. For a lot of experimentalists, it actually felt like the holy grail had finally been found. Why spend months at a crystallography beamline when an algorithm can give you an answer in just minutes? Even the Nobel Academy declared the protein folding problem as essentially solved in 2024 when awarding the prize to Demis Hassabis and John M. Jumper. Not to forget, of course, David Baker as well. But here's the thing. AlphaFold doesn't always get things right. Ask AlphaFold to predict the structure of a protein, such as KaiB, and that's a key player in the cyanobacterial circadian clock, and it often gets things wrong. Not just a little wrong, a lot wrong, like completely wrong, like 5 times out of 5 wrong. And that's because KaiB does something that AlphaFold was never built to expect. It actually switches folds entirely, and it flips between 2 completely different structures just depending on the time of day. So what's actually happening under the hood when AlphaFold makes a prediction? What do these confidence scores really mean? And when should they make you more suspicious, not less? Why does the gold standard in structural biology still run through X-ray crystallography, NMR, and cryo-electron microscopy, even in the age of AI? So in today's episode, we're going to explore the limits of AlphaFold. And hopefully I'll illustrate the key issue that why AlphaFold is a powerful tool in structural biology, it does not generate an experimentally observed structure, and its results should always be analyzed with a little bit of a grain of salt, if you will, right? So let's begin by looking at the systematic accuracy and benchmarking against real crystallographic data. So a 2024 paper in Nature Methods by Thomas C. Terwilliger and Paul D. Adams and colleagues directly compared AlphaFold predictions to experimental crystallographic density maps, not just deposited PDB models, and so they did that to avoid bias. And what they found is that about 10% of the alpha carbon atoms with a pLDDT score over 90% are in error by over 2 angstroms, along with 22% of those with a pLDDT between 80 and 90%, 33% of those with a pLDDT between 70 and 80, and 77% of those with a pLDDT under 70. For comparison, just 5% of the alpha carbon atoms in matched pairs of structures in the protein data bank crystallized in different space groups by over 2 angstroms. And so what this tells us is that even AlphaFold's most confident predictions have roughly double the error rate of two independently solved crystal structures of the same exact protein. And what's especially worthy to note is that even very high confidence predictions differed from experimental maps on a global scale through distortion and domain orientation, as well as on a local scale in backbone and sidechain conformation. And so this tells us that high pLDDT scores do not guarantee correctness. So another case to consider in which AlphaFold gets confused is with some of the proteins that have more than one functional 3D structure. And so we need to remember, first of all, that proteins are not fixed. They're not fixed, they're not solid objects, but they're actually living, breathing biomolecules. As such, movement is a significant part of the function of most proteins, in fact. So let's consider fold switching proteins. And these have more than one conformation and physically switch between two entirely different folds. One excellent case, kind of a textbook case, if you will, is to look at here is KaiB. And KaiB is a cyanobacterial circadian clock protein. In its ground state, KaiB adopts a helix, strand, helix, strand, helix fold. And this assembles into a stable inactive tetramer. Over time, driven by its interaction with the clock protein KaiC, a subset of KaiB molecules slowly convert to an excited state, and this refolds their C-terminal subdomain into a thioredoxon-like strand helix-strand topology. And this excited state fold is the biologically active form. It exposes a surface that lets KaiB bind to KaiC, and this in turn recruits Kia and shuts down KaiC's autophosphorylation activity. This drives a negative feedback loop that generates approximate 24-hour oscillation. So the fold switch isn't just a structural curiosity. It literally is the clock's mechanism for delaying its own signal long enough to produce a full circadian period. And that's exactly the kind of dynamic environment that the behavior of a single static AlphaFold prediction simply can't capture. So let's look at a 2022 paper by Chakravarti and Porter. And so this was first published in bioRxiv. That's the free version. And it was later actually published in Science. So if you don't have access to Science, you should be able to see most of the key information in bioRxiv. And I'll put the description or a link in the description of the video in case you're interested in looking at all these papers and all that stuff. So this paper found that AlphaFold 2 predicts that KaiB assumes an excited thioredoxin fold in 5 out of 5 cases. That's right, yeah, it consistently missed the ground state entirely. More broadly across their benchmark, 94% of AlphaFold 2 predictions captured one experimentally determined conformation, but not the other for these fold switching proteins. And even more interesting is that the estimated confidence was moderate to high for 74% of fold switching proteins, and this contrasts with the overall low confidence scores that are seen with intrinsically disordered proteins. And unlike intrinsically disordered regions where the sequence alignments show fairly low conservation. Fold switching regions had conservation rates that are statistically similar to canonical single fold proteins. So they should fold pretty similarly, you would think. So the work in this paper suggests that AlphaFold's methodology focuses on statistically analyzing the collection of known protein sequence and structural data, but it fails miserably at capturing fundamental biophysical features such as folding pathways. And this highlights the necessity of looking at protein structure not as a static individual structure, but more as a protein ensemble structure. And that's the ensemble is the collection of all structures related to that biological function. So if AlphaFold is ever going to become trusted for use in drug discovery, this is a problem that's going to need to be addressed, because fold switching proteins are used to regulate a pretty wide variety of biological processes associated with numerous diseases. This includes COVID-19, cancer, Alzheimer's disease, even malaria, for example, right? So yeah, we're going to have to kind of solve this problem. A follow-up study by the same authors in Nature Communications, they went even further and showed why. AlphaFold isn't really understanding protein energy landscapes. It's really just simply memorizing structures. So in this new study, they analyzed the fold-switching protein RFAH, which is in Escherichia coli, or E. coli. I love pronouncing some of these words, but Latin is not my thing. Anyway, everybody knows what E. coli is, right? Okay, so RFAH is a bacterial transcription factor, and the C-terminal domain reversibly switches from an all-alpha helical ground state to an all-beta sheet excited state upon binding RNA polymerase and a specific DNA sequence called OPS or OPS. Both conformations of E. coli RFAH have been determined experimentally. And there's also circular dichroism evidence as well as NMR evidence for switching in all 5 of these sequence-diverse RFAH homologs. So all these sequences, less than 35% identity to one another's, as well as to E. coli RFAHs. So it's pretty low sequence identity. Now, AlphaFold 2 has likely memorized RFAH's helical bundle conformation, though, during its training set. And so predictions consistently resemble the helical structure likely in AlphaFold 2's training set. And so when tested on the isolated RFAH C-terminal domain, these predictions contradict experimental observation. And that expressed in isolation, the RFAH CTDs, the C-terminal domain, folds into a beta sheet structure, not a helical bundle. So here we see that AlphaFold can be confidently and consistently wrong in a way that looks like real biology, but in fact it actually isn't, right? So another striking observation in that same paper involves human XCL1, and this is also known as lymphotactin. And so lymphotactin is a small signaling protein in the chemokine family. It's mainly produced by activated T cells and natural killer cells. It attracts immune cells, particularly dendritic cells and NK cells, to sites of infection or inflammation by binding its receptor XCR1. And this helps coordinate the immune response. So XCL1 is a pretty well-known metamorphic protein, or fold switching. It kind of changes conformation, and it naturally interconverts between two completely different folds in solution. One is a chemical chemokine fold and with typical three-stranded beta sheet plus alpha helix, and the other an all-beta dimeric fold. That's a dimer, so that, that's two chains, and that's unrelated to any of the other known chemokine structures. So each fold has a distinct function. The chemokine fold binds and activates the receptor XCR1, and the dimeric fold is needed for binding glycosaminoglycans on cell surfaces, which helps establish the chemical gradients that immune cells follow. And so this is kind of similar to KaiB, where this dual fold behavior makes SCL1 a natural stress test for machine learning structure prediction. And that's also part of why AlphaFold-based models have struggled to correctly predict its dimeric conformation. So for this protein, neither AlphaFold 2 nor AlphaFold 3, we've got two of them now. We're looking at both of the major methods. Neither of these successfully predict the dimeric conformation of human XCL1. AlphaFold 3 predicts an experimentally unobserved domain swap structure over 16 angstroms, and that's different from its experimentally observed counterpart, despite having reasonably high confidence scores. So, the confidence score in AI kind of tells you how confident it is with its structure, but confidence does not necessarily mean accuracy. That's an important distinction, right? We don't want to get those two confused. It just means the AI is confident, but it can still be totally wrong. Now, a 2025 study by Molnar and coworkers, and this is published in the Computational Structural Biology journal. This looked at nuclear receptors, and they observed several domain-specific weaknesses. They found that AlphaFold actually does pretty well in rigid domains, but it shows limitations in capturing the full spectrum of biologically relevant states, particularly in flexible regions and ligand binding pockets. These include ligand binding domains that show higher structural variability. The coefficient of variation is 29.3%, and this compares to the DNA binding domains with a coefficient of variation of 17.7%. And so what this illustrates is that AlphaFold accuracy is not simply one number. It actually depends heavily on what part of the protein and what kind of structural element that you're asking about. So these fold switching studies make a very important conceptual point. And that point is the deep learning-based structure prediction methods are likely to miss protein properties that are not apparent from available experimentally determined structures. And furthermore, AlphaFold 2 predictions cover 99% of sequences in the human proteome, but only 58% of the residues are modeled with high confidence. This is actually a great reminder that AlphaFold has predicted the whole proteome, and AlphaFold has solved the whole proteome, are two very entirely different claims. So prediction and actual solution, two completely different things. So I think it's fairly imperative to consider and redefine what AlphaFold actually is, especially since it has apparently seeped into wet lab culture as if it's the gospel of ground truth. We need to consider and remember that AlphaFold is a hypothesis generator, not a structure. AlphaFold predicts what a protein probably looks like. And that's based on patterns in known structures, what it observes in its databases and the 3D database as well as the sequence database. It doesn't actually observe your protein. Experimental methods such as X-ray crystallography, NMR, cryo-Electron microscopy, these actually do observe your protein. Even circular dichroism, these actually observe structural characteristics of what your protein actually does and what it actually looks like. So as with any AI method, you have to remember that AlphaFold is actually trained on and what it's optimized for. It's pattern matching against the protein data bank and evolutionary covariation. It's not physically simulating folding, and it's certainly not measuring anything. It has no way to know about your specific construct, ligands, post-translational modifications, oligomeric state, crystallographic solution conditions. Doesn't have any of that data at all, unless those patterns actually exist in your training data. It's only as good as the training set, as with most AI methods that are on the market today. That applies to generative AI as well. And so keep in mind that the predicted local distance difference test, that's the pLDDT, and the predicted aligned error, PAE, are not the same thing as accuracy. And certainly not in the experimental sense. These are computational constructs. So a high pLDDT score, that simply means that the model is confident that this matches the patterns that the AI has seen before. It does not mean that matches your actual protein. Low confidence regions, these can include flexible loops, amine and carboxy terminal regions, disordered regions. That's often exactly where the real biology often happens. And so AlphaFold also struggles predictably in certain cases, especially with conformational ensembles, multiple states, ligand-induced conformational change, protein-protein interfaces without good multiple sequence alignments, intrinsically disordered regions, novel folds with no evolutionary relatives, and even effects of mutations, post-translational modifications, and bound cofactors. AlphaFold 3 does show some improvement in some of these areas, but not without a certain margin of error. It's still generative AI. It does work by a slightly different mechanism than AlphaFold 2. It's better in some areas. It's not as good in other areas. Either way, whether you're looking at AlphaFold 2, AlphaFold 3, or Rosetta Fold, or any of the others, it's still just a prediction or a hypothesis. It's not an actual structure, and we need to remember that. We also need to remember that proteins are dynamic ensembles. And so AlphaFold gives you just one snapshot, or maybe about 5. I think, it's common to get about 5 snapshots. But it doesn't capture the entire conformational landscape the way NMR ensembles or multiple crystal forms can. And of course, X-ray, NMR, and cryo-EM each have their own caveats too, right? And, handling and accounting for error and observation is, of course, one of the fundamental hallmarks of good science. Experimental error can include things like crystal packing artifacts, resolution limits, cryo-EM map interpretation at moderate resolution, those sort of things. So experimental methods are certainly not infallible, but experimental errors are of a fundamentally different and better characterized kind of error than those in predictive modeling that we see in machine learning methods. And we even see this in generative AI as well, too. Everybody's seen some pretty bad AI-generated pictures and stuff that is just completely out of left field. We know AI is certainly not always right, so let's keep that in mind even in our science as well, too. So let's conclude this episode with some practical guidance, and especially for experimentalists, but even anyone working with machine learning-based structured prediction, and this includes computational scientists too. I'm not going to just pick on the experimentalists. First of all, you always want to check the pLDDT and the PIE error plots. These are not just the cartoon structure, so just always look at all of your data that comes out of the AI. Secondly, cross-check the predictions against any existing homologous structures, such as homology modeling. We want to superimpose that AI prediction against any known experimental model or a similar structure, right? We also want to treat AlphaFold models as a starting hypothesis for experiment design, such as mutagenesis targets, construct boundaries. And keep in mind that it's not a final answer or for the mechanism or for any publication level claims. We're just making a prediction here, and we need to keep that in mind. We want to be especially cautious using AlphaFold models for binding site druggability or docking without actual experimental validation as well. So this is going to be very important moving forward, especially the active site has a lot of variability. It's got a lot of movement. It's got a lot of factors that are involved there. There can be cofactors involved. There can be allosteric implications, that sort of thing. And that's hard to model and it's hard to predict. We also want to know when a question requires experimental structure determination, such as conformational change, ligand binding poses, dynamics, etc. Right? So I guess today I've probably been a little critical of AlphaFold, and you're probably thinking, yeah, let's not use it. It kind of sucks, right? While it's important to take any AI predicted structure with a rather large grain of salt, it's still an incredibly useful tool for computational and experimental scientists alike. And so I hope that you now see this as an important tool in studying protein structure and providing hypothesis for further experimental testing, as opposed to some godlike decree of, hey, this is the absolute structure right here. This is what it actually looks like. It's not perfect, but like any experimental method, it can provide a lot of insight into your protein, a lot of insight into the biological function. So we should still use this, but like anything, we have to consider all of the possible errors in our methods and not just take one thing for granted. So once again, thank you for watching Biochemistry Unfolded, from atoms to algorithms. And if you enjoyed this episode and found it interesting and insightful, go ahead and hit the little like button down there. And if you want to see more interesting content, subscribe to the channel, hit the little bell icon, and that way you're updated whenever I post new episodes and content. If you have any questions about AlphaFold or anything else, please leave them down in the comments below, And, I read those comments as well, and it kind of tells me who's reading and who's watching the stuff that I'm posting here. So that's always very helpful to get those interactions. And again, I will see you in the next episode. Thanks for watching. You have been listening to Biochemistry Unfolded, from atoms to algorithms. Thank you for watching. We will see you again in the next video.